Pathology. Urethral gonorrhea begins as an acute inflammation at the meatus, whence it travels inward along the urethral mucous membrane. Unless repressed by local treatment, this inflammation invariably travels as far as the bulbous urethra and usually invades the posterior urethra as well. Although gonorrheal inflammation is essentially the same in the anterior as in the posterior urethra, we shall describe these processes separately. The cytology of gonorrheal pus has been studied by Joseph, Weil, Posner, Neuberger, and Taylor. The results obtained are nil. The predominating cell is polymorphonuclear. The eosinophil is always encountered but does not harbor gonococci and is not pathognomonic. As Taylor says, all of this detailed study of cellular elements of gonorrheal discharge, interesting as it may be, has contributed very little to our understanding of the nature, prognosis, or treatment of urethritis. Pathology of acute anterior urethritis. Onset. The most accurate data we possess in reference to the invasion of the urethral tissues by the gonococcus are those published by Finger, Gohn, and Schlaugenhaufer. These authors inoculated the urethra of criminals condemned to death and were able, by means of immediate post-mortem examination, to investigate the various stages of invasion of the tissues by the specific microbe. 38 hours after inoculation, the gonococci had only just begun to effect an entrance between the epithelial cells. The lacuna of Morgagni was crowded with the cocci. Diapedesis had begun, and intracellular gonococci were found among the few leukocytes on the surface of the epithelium. At the end of 3 days, the inflammatory process was well underway. The surface of the mucous membrane was covered with pus, its epithelium infiltrated by bacteria from one side and by leukocytes from the other. The inflammation showed 4 striking characteristics, viz. 1. The pavement epithelium of the fossa navicularis, although swollen with leukocytes, resisted the invasion of the gonococci almost absolutely. 2. The cylindrical epithelium of the penile urethra was generally invaded. 3. This invasion was most marked about the crypts and glands, which were packed with pus and gonococci. And 4, the subepithelial connective tissue, though showing every evidence of inflammation, contained few gonococci, except in the neighborhood of the crypts and glands. Height. When the inflammation reaches its height, the mucous membrane from the tumified meatus to the bulbous urethra is intensely inflamed. Its surface is red and swollen, covered with pus, and, in places, eroded or ulcerated. The epithelia and the subepithelial connective tissue are infiltrated by the warring gonococci and leukocytes. The glands and crypts form the most important centers of inflammation. They are implicated in the general process, inflamed and distended with gonorrheal pus. The gland orifices become obstructed by the tumefaction of the mucous membrane, and the glands thus form centers whence infiltration and even suppuration extend into the surrounding tissues. Since the glands are numerous and extend to, and in some instances, even into the corpus spongiosum, the submucous infiltration or suppuration arising from them is often widespread and intense at the height of an acute gonorrhea and leaves infiltrations that are the chief cause of chronic urethritis. Were the urethra as glandless as the conjunctiva, urethral gonorrhea would show as little tendency to chronicity, complications, and systemic absorption as does conjunctival gonorrhea.